All right, I just wanted to make a quick little video that helps understand a principle known as Le Chatelier's principle. And this is a really important concept that for some reason some people just struggle with when it comes to understanding, among other things, acid-base balance. Of course, this principle is applicable to any reversible chemical equation, and it's really, really useful in understanding how buffer systems work, in particular, the bicarb buffer system that we talk about so much in anatomy and physiology is a significant part of the buffering capacity of your body. So to just lay the groundwork for this principle, I'd like to start off with just a simple teeter-totter. It's important to understand, when we talk about Le Chatelier's principle, the whole concept of equilibrium. And when it comes to this teeter-totter, I think it's pretty straightforward and makes sense that equilibrium means balance. In other words, if I have something on this side that weighs more than something on this side, we know what's going to happen. The teeter-totter is going to swivel in that direction. There are two ways that we could actually bring the teeter-totter back into equilibrium. We could add more weight to this side, even though they're not quite the same size, but we could add more weight, and that's going to restore equilibrium. Alternatively, what if we changed where the fulcrum was? If we were to shift the fulcrum to the left, now we have equilibrium, balance, even though the two sides don't have the same amount of stuff. And this is kind of an important concept when we think about what equilibrium actually means. Equilibrium is simply a balance in the system. There's lots of ways to achieve it, but by nature, reactions will always strive toward that equilibrium. Now, it is important to recognize, if we're talking about chemical reactions, equilibrium needs to be defined as the rate of forward and reverse reactions being the same. It's important to note, this says nothing about concentration. It does not mean that the concentrations of products and reactants are the same. It just means that they're in equilibrium with each other. In other words, the products are turning into the reactants at the same rate that the reactants are turning back into the products. That's the idea of equilibrium in a reversible reaction. Now, if we apply this to the acid-base system, let's give an example of a generic acid-base equation where we have a weak acid, HA. We know it's a weak acid because it's a reversible reaction, which means it does not dissociate completely, but it will dissociate partially, giving us a population of H plus and A minus. We can define these. This is our weak acid. Of course, these are the donated protons. H plus, hydrogen is just a proton and electron, but this is ionized, it's lost its electron, and so what do I have left? I have a proton, and that's what we're actually measuring when we think about pH, the concentration of protons. And then, of course, A minus, what's left over after the dissociation of the weak acid. This is now the conjugate base, conjugate meaning its pair. Right? It's not just a random base, it's the pair to the weak acid. Or if I had a base, I would have its conjugate acid. They're paired. And that's what we need to have a buffer system. So now we want to take this generic equation and see what happens when we put stress on the equation, right? That's the idea. Going back to the idea of equilibrium and the teeter-totter, if I add weight to one side, what does it do? It shifts equilibrium. The teeter-totter becomes out of balance. What happens naturally next is what the reaction spontaneously does to achieve a new equilibrium. All right, so let's get back to this idea where we've got HA, and it is in dynamic equilibrium with H plus A minus. The question is this: what happens if we stress the system? In other words, what happens if I increase the amount of weak acid? If the amount of weak acid, HA, goes up in the system? The best way for me to think about this is going back to these arrows right here. If we can imagine these arrows as vectors that help us understand the rate of the reaction, right now the two arrows are the same size. But what would happen if this arrow was big compared to this arrow that's small? I hope you can appreciate that in that scenario, the overall direction of the reaction would be toward the right. Now, let's see if we can't understand what's causing the whole size of the arrow to help us make this comparison. If we look at HA, really HA consists of, as we can see, it would dissociate into H plus A minus. It consists of two parts. It consists of the H part, which we'll symbolize with a yellow dot, and the A, which we'll show with a green dot. These things are currently bound together, but will fall apart during dissociation. They fall apart at a specific rate. When we think about interactions at the molecular level, in this case, this reaction, it's not a very stable interaction. In other words, it's a boyfriend and a girlfriend. They kind of like each other, so sometimes they hold hands, but sometimes they're just going to let go, right? They go out in public, I don't want anyone to see me here, so they let go. And when that happens, what do we see? We see a dissociation between the two. So we see the yellow not holding hands, but now independent of the green. Now, the beauty about this chemical reaction is that we can predict the tightness in which these two molecules are holding hands. So the idea behind equilibrium is a balance between these two systems. In other words, at equilibrium, the ratio of the products and the ratio of the reactants is the same. This is actually called the equilibrium constant, K equilibrium, which simply tells me that the concentration of my products and the concentration of my reactants maintain a stable relationship, a ratio. So, let's say, in this given scenario, we show 5 pairs. And it's in equilibrium right now where we have five pairs or 5 green-yellow combinations representing our weak acid, and it dissociates a little bit into 1 yellow and 1 green. And so we see the ratio defined here as 5 to 1. And in this ratio, for this given system, the arrows are the same size. In other words, for every 5 pairs, one of them decides to let go of each other, and we have 2 independent, lonesome souls. And what these arrows right here mean is that these lonesome souls are deciding to hold hands again and join the couple side at the same rate that one of these couples is deciding to let go and join the lonesome side. So what's going to happen if we add, let's say, 5 more pairs? That gives us 10 in total, the 5 that we started with, plus the 5 that we just added. If the rate is the same, I hope you can appreciate that now, because I have twice as many couples, the odds of one of them letting go are twice as high. That arrow becomes bigger. We haven't changed anything over on the single side, and so that arrow stays the same. if we add those two arrows up, what do we see? We see a net movement toward the other side. We know, because we're trying to maintain that same ratio, that we're going to end up with another single yellow and another single green. So now, where we have 5 plus 5 gives us 10, now we have over on the right side 1 plus 1 to give us 2. Importantly, the ratio is the same, right? This is still a 5-to-1 ratio. Equilibrium is maintained. Well, let's consider a different scenario where instead of adding more of the weak acid, the couples, what if I added more of one of the singles? What if I added a bunch more of these yellow dots? I didn't change the concentration of the green dots, right? I still have just the one. And over on the other side, we'll separate, maintaining good separation. Over on the other side, we'll go back to what we started with originally. We have 5 couples. Well, we haven't changed anything about the couples, right? So now in this scenario, the arrow going to the right is the same. But if we look at what we did to the singletons, remember, the whole reason they're turning into a pair, is because they decide to hold hands. They find each other, they like each other, they decide to hold hands. In this scenario, by adding an increase in the yellow singles, we've significantly increased the odds of one of those yellows finding the green dot. So what have we done? We've increased the size of the red arrow, or the bottom arrow, going the other direction. In this scenario, if we add these two arrows up, we see a net movement in that direction, to the left. So for this given scenario, we are going to see an increase in what's being produced, because this is what this arrow tells us, right? It tells us what's being produced. We see an increase in what's being produced, which is, in this case, a couples pair, and a decrease in what's being consumed, right? That green dot bound to a yellow dot to turn into a couple. And it did that at an increased rate because we added more yellows to increase the odds of a collision between the yellow and the green dots. That's the idea of Le Chatelier's principle. If I disrupt equilibrium, I need to be able to predict how the equation is going to respond and adapt as it seeks a new equilibrium. But remember, the equilibrium is just the ratio of products to reactants. It likes to be, or it seeks out the point where the forward and reverse reactions are happening at the same rate. So let's take this and let's apply it to the actual acid-base buffer system, the bicarb buffer system, that is the most prominent buffer system in your body, in the blood. So for the buffer system, the weak acid, carbonic acid, H2CO3, is in dynamic equilibrium with H+, and bicarb ion, the conjugate base, HCO3-. Now, something interesting about this system is it's also in dynamic equilibrium with carbon dioxide and water. In other words, carbon dioxide plus water will turn into carbonic acid. All of these things are in equilibrium with each other. So for our purposes, I'm going to take out the intermediates. We're not going to worry too much about that. And of course, the body's made of 65% water anyway, so I'm not going to worry too much about that. And we're simply going to write out the equation like this. Carbon dioxide is in equilibrium with H+ plus HCO3- bicarb ion. And what we want to be able to do is determine, we've got 6 different possibilities for ways that we can stress this system. we can increase carbon dioxide, or we can increase hydrogen ion, or we can increase bicarb ion, or we can flip the whole thing around. We can decrease carbon dioxide, we can decrease hydrogen ion, or we can decrease bicarb ion. These are 6 different ways where I could put stress to this equilibrium in my body. And our job for each of these is to predict what happens and understand how the other components are going to adjust. So the first thing I want to do is I want to figure out which direction the reaction is going to go. Remember, from our discussion up on top, let's start with this first one right here. If I increase CO2, this is kind of like increasing the couples, right? And so what am I going to see? I'm going to see an overall net drive to the right. And because this arrow tells me what I'm producing, I'm going to predict an increase in H plus and an increase in HCO3. As carbon dioxide dissociates, it will eventually turn into H plus HCO3. This is the reaction seeking a new equilibrium and describing the dissociation of carbon dioxide. In this case, it's binding to water, turning into carbonic acid, which then dissociates into bicarb ion and H plus. Likewise, if we go down to the next one, number 2, what do we see? We see an increase in the singles. This is going to push the reaction to the left. This tells me that carbon dioxide is going to go up at the expense of bicarb ion, which is being bound to H plus in order to make CO2. And likewise, if we see as a stress, the increase in bicarb ion, again, that's going to force the reaction to the left. We're going to see an increase in CO2 and a decrease in H plus. Now, for all of these, one thing that's really, really easy to note, if the arrow is up arrows, and I'm talking specifically about the stress, what's shown in yellow, right? If the arrow is up arrows push away. And so you can see as this goes up, it pushes away. It pushes in that direction. As this goes up, it pushed in that direction. That's an easy way to remember how to do this. And now we'll see on the bottom end, down arrows pull toward, right? Because this is going down, it's going to pull the arrow, the reaction toward itself to replace what was lost. And we're going to see, because everything on the right side now becomes the reactants, we're going to see a decrease in the reactants. Likewise, here the yellow is pulling down, so it's going to pull the reaction towards itself. We're going to see a decrease in the reactants and an increase in the products. And we're going to see that same thing here. We get pulled to the right. We're going to see a decrease in the reactants and an increase in the products. Now, for physiology students studying acid-base balance with the kidney and with the lung, there's a couple of things that I want to point out. Of course, carbon dioxide is regulated by the lung. H plus and bicarb ion are regulated by the kidney. 2 key things to note. Whenever we see a yellow arrow on this side, in other words, when carbon dioxide is the problem, we see that the arrows on the right side are pointed in the same direction, right? We see that in both of these cases. Whenever the arrows are pointed in the right, these arrows are pointed in the same direction, this is a respiratory problem. Is it respiratory alkalosis or respiratory acidosis? All we have to do now is look at the H plus, right? In the first one, we see the H plus is up, which means that pH is down. I'm becoming acidic. That's going to be a respiratory acidosis. Whereas in the second one down below it, we see decreased H+. That's going to be a respiratory alkalosis. In the other four scenarios, we see these arrows going in opposite directions. And in all four scenarios, the yellow arrow is in one of those pink circles, right? So whenever we see the arrows of H+ and HCO3- going in opposite directions, we know it's a metabolic problem. And then again, depending on the H+ concentration, if it's going up or down, we would diagnose it as a metabolic acidosis or a metabolic alkalosis. And then it's just important to recognize the compensatory mechanisms that the body has in place to try to deal with it. In an oversimplification, if the kidney's the problem, then the lungs can help out. Or if the lung's the problem, the kidneys are going to try and help out to maintain that homeostasis. As an example, let's look at this scenario up here, number 1 again. Okay. What would cause an increase in carbon dioxide? Well, this is something that we would consistently see in something like COPD, right? Where someone, maybe we've got a smoker or a coal miner. Long term, they've damaged their lungs and their ability to ventilate is significantly compromised. And so mechanically, they're struggling to breathe off all of the carbon dioxide. So what happens? The stress to equilibrium is an increase in carbon dioxide. CO2 levels go up. So if we were to draw that out, there's my stress to the system. CO2 levels go up. We know what's going to happen. We know that the body will seek a new equilibrium, and because this is an up arrow, it's going to push away. So we know that we're going to have a shift to the right, and we're going to see an increase in H+ and an increase in HCO3-. This specific set or scenario can only describe respiratory acidosis. Now, what's the body going to do to try and compensate for this? Because it's a respiratory problem, it's a chronic problem, the body's going to try and respond with the kidneys. And so in this case, because it's acidosis, the kidney is going to activate the type A cells of the intercalated cells in the collecting duct. And the job of those cells is to pump acid into the filtrate and to pump bicarb into the blood. And so the compensation is going to see an increase in bicarb ion. This is what the kidney's doing to fix the problem. Now, if that's the stress to the system, consider what happens. That's going to push the reaction in the opposite direction. I'm going to see an increase in my products, and now it's going to be CO2, but the concomitant decrease in H+ concentrations. Compensation just means I'm going to take both of these scenarios now and I'm going to add them together. And what do we see? We see double arrows, 2 pointed up for CO2. For H+, one's pointed up and one's pointed down. And for bicarb, they're both pointed up. So this is what compensation does. It takes these two arrows right here. They cancel each other out. And so even though the individual has a normal pH, I know that it's being compensated for. I know that it's a metabolic acidosis and that it's being compensated for because I see massive levels of bicarb in the blood, and I also see elevated PCO2 levels. But at the end of the day, the fact that my pH is, for the most part, normal, this shows how the body is compensating for a problem of elevated CO2 without having the disruption that can be caused by alkalosis or acidosis in the body.